Hello everyone, my name is Lucas Salles Queiroz. I am a PhD student from Federal University of Viçosa in Brazil. And part of my PhD, I work as a guest PhD student in Denmark at Technical University of Denmark, DTU. And today I'm going to present a work that was developed last year at DTU based on the physical and interfacial properties of black soldier fly protein extract before and after hydrolysis. So this is the work. The work was done in collaboration with Angelique Berton and also under the supervision of Mohamed Amin Mohamed Far, Antonio Fernandes de Carvalho, and Federico Casanova. So in the first part here, we see the physical chemical properties of a protein extract of black soldier fly and the hydrolyzed sample from black soldier fly. So in this part, I will show you the results of protein content, zeta, potential, size, and surface hydrophobicity. So in the beginning of the project, I started extracting the proteins by the chemical extraction, which is a conventional common process where we obtain the proteins by the electric point. And then after that, we obtain the protein extract, and then they go through the hydrolysis. And for that step, I use alkylase. The degree of hydrolysis that I chose in this process was 4% and 12.5%. So here, the first data we obtain is the protein content. As you can see here, the black soldier fly, the whole proteins show the protein content of around 70%. The degree of hydrolysis 4% was 67% around, and 12.5% was around 65-66%. And the first analysis I did in this protein was the surface hydrophobicity. So the surface hydrophobicity is important to understand how this protein will interact with the oil phase, for example, in case we work with emulsion. So how it can perform, like can stabilize better an emulsion and bind to the oil phase, for example, based on the presence of hydrophobic amino acids and hydrophilic ones. So if we have a well distribution of these hydrophobic groups, the chances are higher to stabilize better a droplet, for example, in an emulsion. So in this case here, we have surface hydrophobicity of 130 for whole proteins before hydrolysis, and after hydrolysis of 4% and 12.5%, we see a decrease in the surface hydrophobicity. And this, we came up with a hypothesis that maybe as we hydrolyze more, we obtain small peptides, and the small peptides started aggregating and hiding and burying these hydrophobic groups. As we will see here, when we check the size of the proteins and the aggregates after the hydrolysis, we can see here that whole proteins show it intermediate size, 140 nanometers, that's the diameter. And the degree of hydrolysis 4% showed the smallest aggregates, and the biggest aggregates was performed by the 12.5%, which corroborates with our hypothesis that these hydrophobic groups are maybe hidden inside of these aggregates. So that's why we have lower hydrophobic surface and also a bigger aggregates in the 12.5%. So from now we analyze the interfacial properties of proteins before and after hydrolysis. So the first data we have here is the interfacial tension, which we analyze using a pen and drop equipment. Basically, we have a droplet form in the very tip of the syringe, and then it's placed inside of a cuvette with oil, and then we have the migration of proteins in this water phase going to the interface where we have the contact between oil and water. And then the idea is that we can reduce the interfacial tension as we increase the number of proteins in this interfacial area. So we can see how fast the protein goes to the interface and how they are able to decrease that in a graphic of interfacial tension by time. So as you can see here, it was analyzed for more than 1 hour. here, and in the green line, we can see the water phase is just control that we don't have any proteins, and interfacial tension was around 24, and at the end reaching around 22. When we have here in the blue, red, and green, we have blue the 12.5%, red is the 4% degree of hydrolysis, and black the whole proteins without any hydrolysis. And as you can see, the whole proteins were able to go faster to the interface and also reach lower interfacial tension after all the experiments, after 80 and more, 90, 85 minutes of analysis. So here we came up that as BSFL whole proteins without any hydrolysis has a higher surface hydrophobicity. The hydrophobic interaction between the oil and water was more interesting to stabilize better, and then we could create a better net and stabilize more and reduce more the interfacial tension between the water and oil. So that's one of the hypotheses we have. Also, degree of hydrolysis 4 shows the smallest size, so they were able to go faster to the interface compared to the 12.5%. That's why we have here the red line in the middle. So the next experiment we performed to understand interfacial properties is the shear rheology, which is a little bit different. So as you can see here on the right side, we have a double wall ring, which is the method we use, which is basically we have water and then we have oil, and then we place this double wall ring in the interface between half in the water and half in the oil. And then the idea is that we can fill by this the oil, by this ring, actually the proteins migrating from the bottom from the solution, the water solution, to the interface as they go directly to the oil phase. And then based on the hydrophobic groups and their interaction, the peptide size and everything, we can see how they migrate to the interface. And then when we have more migration to the interface, we have an increase in the G prime, which is shows the elastic behavior of the sample. And when we have a lower G prime, then we normally have a higher G double prime, which is related to the viscous behavior of the sample. So as you can see here, the whole proteins since the beginning of the experiment shows a higher G prime, which is more elastic behavior. And we have an oscillation here, probably because we have the formation of this net protein net that was destabilized at some point, then broke down, the proteins were dispersed in the median, and then they start forming again because of the higher number of hydrophobic groups, as we analyzed before. And when we compare degree of hydrolysis 12.5 and 4%, we can see that take a longer time to have G prime higher, which means that took longer to the proteins go to the interface and form a stable net there and stabilize this interface between oil and water, and then perform a more elastic behavior. So you can see here it took 19 hours and 24 hours for degree of hydrolysis 12.5 and 4% respectively. So in the last part, we analyzed the emulsions, which was made with sunflower oil in water, emulsion stabilized with BSFL, proteins, whole proteins, and hydrolyzed proteins. So in this test, we used TurbiScan to analyze the physical stability over more than 50 hours. And then we used the droplet size and the zeta potential of the droplets to try to understand better how the emulsion will behave during storage process. So emulsion physical stability, as you can see here, the analysis was performed in TurbiScan, which is like a tower where we insert the samples there, and we can just leave and analyze the backscattering and the transmittance. And based on the backscattering and the transmittance that we obtained in data, it creates a general data and a general value which is called TSI. That comes from the company that made it as equipment. And when we have TSI around 3, it means that the sample is very stable. When it's higher than that, then the sample starts getting unstable. So as you can see here, after 15 hours of experiment, we still have here a stable sample for both the degree of hydrolysis 4 and 12.5, but not for BSFL whole proteins as they were. It was higher, the stability was lower since the beginning, as you can see here. And if we check the degree of hydrolysis 4, as we also saw the smaller proteins before and higher stability here, we can see that it goes more stable even at the end of the experiment, and which is not true for 12.5%, as we have a higher increase here. And if we kept the experiment for more days, we could see that the 12.5 tend to increase and get closer to BSFL proteins. So that's the general physical stability over 50 hours of experiment. So for that, during each day, during the more than 48 hours, we analyzed the droplet and the size of the sample. So we can see here, day 0, we have smaller droplets for both hydrolyzed sample, which was very similar. And as we can see here, it was very similar in the beginning for the stability, but not for the BSFL, which showed a much higher and significant different droplet. So here we can see as the time goes from day 0, day 1, and day 2, we have a slight increase, but always higher BSFL whole proteins compared to the degree of hydrolysis 4. But for 12.5%, we have a higher increase, a significant increase, different compared day 0 and day 1 and then day 2. So which corroborates with the data we have here for the physical stability. As you can see, it increases much more compared to the degree of hydrolysis 4. So smaller diameter for BSFL protein hydrolyzed at day 0. That is what we can see in both graphics. The degree of hydrolysis 12.5 has the biggest diameter at day 2, whereas the degree of hydrolysis 4 increases less through time as time goes. Okay. BSFL protein droplets were bigger and slowly increasing through the time. So as the time goes, we can see that it didn't increase that much, was always higher compared to the degree of hydrolysis 4. And here we have the droplet size distribution, which also meets the same idea as we discussed before. We have a slow increase for BSFL protein, but not for 12.5. And also the polydispersity of these samples, we have very polydispersed sample, like as you can see here, we have a formation of around 10 micrometers droplets at the end of the experiment, and the same thing happened for degree of hydrolysis 4, and more intense increase for 12.5, which corroborates again with the TurbiScan data. So in conclusion, with these results, we have the enzymatic hydrolysis with alkylase could be an alternative treatment in order to improve emulsifying properties. It opens the possibility to test different degree of hydrolysis and also test different enzymes and compare with the data herein reported. So the combination of hydrolyzed sample with other emulsifier can be also a possible sustainable approach for next studies. So in this study, we aim to discover how these different degree of hydrolysis could affect the stability of emulsion when you use BSFL proteins as emulsifier, but also we can maybe further use these emulsifier with other dairy proteins, for example, or other proteins that might show a more interesting stability, physical and maybe oxidative stability for emulsions. So it has been shown in this study that by applying limited degree of hydrolysis, the stability of oil in water emulsion with sunflower oil, it was enhanced, and it was thanks to the BSFL protein hydrolyzed used as an emulsifier. So it doesn't mean that if we increase too much the degree of hydrolysis, we will have a much better stability. As we could see here, the stability was more interesting when we have a lower degree of hydrolysis, around 4%. So I would like to thank you for your time and attention. This is part of a work. We have more data. We are still running some experiments for this project and that we are to publish soon. And this is part of the data. And if you have any questions, I've been working with a sustainable source of proteins during my PhD, and I have also more two papers published with black soldier fly. So if you have any questions or would like to discuss more, we can just reach me by the email that I have here in the slides, and I'll be glad to help you. Okay, thank you very much.